Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm really excited to be here with Dr. Jagesh Shah, who's the Chief Scientific Officer at Mirai Bio. This is a conversation I've been wanting to have for a long time because if you listen to this podcast or if you're in the field, you have probably heard me talk about delivery as one of the biggest challenges in gene therapy and advanced therapies more broadly. And Jagesh has spent a major chunk of his career initially as a systems biology professor at Harvard Medical School and then moving into industry working on this problem. And it's the focus of Mirai. So we're going to have a great conversation about it today. Thank you for taking the time to join me.
Oh, well, thank you for having me, Patrick. It's real pleasure to be here, and I love to talk delivery.
For people who are not as in the details, I'd love actually if you could just start by framing the challenge. When people say delivery is the bottleneck to gene therapy or advanced therapies, what exactly are they talking about, and what's your take on the big problems?
Yeah, so maybe I'll just reframe this as the nucleic medicine era that we're in now really solves a problem that I thought about quite a bit all over my career is when you identify a molecular pathway and you want to intervene in a disease, you often have to go to what I always call the medicinal chemist that went to Hogwarts. Right? This is a remarkable transition because you're taking your biology that's in the language of proteins and nucleic acids, and now you convert it into small molecules. And this has been a remarkable, opportunity for drugs. So I don't want to dismiss that, but the ability to be able to deliver the language of cells has been kind of a transformative element that I've been trying to push for a long time. And so the real question is, what we're trying to do is deliver nucleic acid so that we can reprogram or complement a mutation in a cell. And we're packaging the nucleic acid because the nucleic acids themselves would be degraded by the body. And this is indistinguishable from a viral infection in some respects. So, hundreds of millions of years, the human body has been pushing out foreign nucleic acids to prevent that from happening. And so our job is kind of to engineer the delivery system so it stealthily delivers the nucleic acid. And that's the challenge. You're not just trying to understand the chemistry or in some cases the viruses that you can leverage for delivery. You're also trying to overcome hundreds of millions of years of evolution that have prevented nucleic acids from entering cells because that's something that has historically been bad from the point of view of a viral infection. But now we want to flip that around and try to make it a therapeutic.
And historically, the most straightforward places to deliver for different reasons have been the liver and the brain, the eye. There'll be others probably that you know better
Right.
than me. Can you talk a little bit about where the low-hanging fruits have been, and then where's the next frontier?
Yeah, so the low-hanging fruits have historically been areas that you can access either by putting it in the bloodstream and a lot of it goes there, or directly accessing, kind of that compartment. So again, if we talk about CNS, you have access through intrathecal or intraventricular. So these are the structures that house the CSF, which is essentially, the blood of the CNS. So if you can get it into that liquid, then can you get access to the cells of interest? The eye is an area that I think also has the ability to access, whether through the front of the eye, through the vitreous or subretinal. We've developed the techniques to do that. So being able to get in there has been an area of real interest. It also has the added element that there's a view that it's immuno privileged. So that idea that if we put material in there, that you're not going to see inflammation. Unclear. The human data would speak to the fact that maybe it's not as privileged as we
Mm,
think it is. Then ultimately, the liver is a really interesting place because so much of the blood goes there. So much of your blood goes there to be detoxified. And as a result, we can take advantage of the hepatocytes, which basically are cleaning up the blood, and we can make it so that these things look like they should be things to be cleaned up. And then they naturally get taken up by the hepatocytes. So again, you pointed out some really critical areas: the brain, the eye, and the liver as areas. Now, where do we want to go? We want to go to the muscle. So on one hand, we have intramuscular injection that we got for a vaccine. That's not really muscle delivery per se. It's just getting it into the muscle that then it drains into a lymph node, and then we see the expression of, say, the COVID spike protein, so that you can get a immune response to that. I think that where we really would like to get to is delivery by infusion into the blood, or even by sub-Q access to the blood, and then really be able to have these particles home to all of the muscles, or in the case of the CNS, go through the blood-brain barrier. So we don't have to do a direct injection into the brain. These are areas that I think will unlock tremendous potential for therapeutics. Just they would, dramatically increase. We already know, again, this is the area of nucleic acids. We already know what to deliver in many,
right.
many cases. It's just being able to get it there. And the idea that a therapeutic is stymied by delivery is really kind of what the company that I work at, Mirai, is really trying to kind of make sure is no longer a barrier.
Yeah, that's a great segue to actually talk about Mirai and the platform. Can you tell us a little bit about how it works? What's the major focus right now, and what are you all developing?
Yeah, so I guess the word platform means a lot of things to a lot of people, and I think for me, having made the transition out of academia to industry, platform technologies are generally areas where you solve, a generalized scientific problem, and it applies to many different therapeutic outcomes. And here, what we're trying to do, and I think you framed it well, is we're trying to build delivery systems that can be modularized so that we can take them and send them a lot of different places, simply by just changing a few components. So that kind of rational design, I think, is one critical piece of what we're doing. In addition, we are trying to figure out how to get to these really hard to reach tissues. So we have a core element, which is we have really good liver lipids. So as you pointed out, the lipids, again, the ionizable lipids is kind of the linchpin of the LNP. So that mixture, getting good ionizable lipids that go to the liver, still is a problem. We still want
Yeah.
to be able to get to liver. Getting to immune cells, it's an area of great interest. I think probably the leader here, the tip of the spear, is really in vivo CAR T, being able to take those ex vivo products that have been remarkably powerful and then move them so that we can reprogram in situ. Then I think, we're talking about other organs: lung, pancreas, kidney, brain, muscle. So those are the harder ones, and that's where Mirai has spent a lot of its time trying to figure out how to, what I would call, broach the endothelial barrier, so that we can give it by IV, and these things will know to home to a particular tissue and a particular cell type, and deliver the genetic cargo that of interest. And again, we've focused entirely on delivery, so we don't make any therapeutic assets of our own.
Right.
And so this is maybe a different, business model than what we might have encountered in the past. But the goal is, people really have good nucleic acid ideas out there, and our job is to enable the delivery to the sites that they want to be able to access.
And your focus, you mentioned the ionizable lipids.
Right.
Your focus is lipid nanoparticles. I would love if you could talk a little bit about lipid nanoparticles
Right.
versus, viral vectors, other strategies that you have considered or might consider. Also, just to help people understand, like, delivery is not one, even one. We've got boats, we've got planes, we've got cars. There's a
Exactly.
lot of different ways to deliver.
There's a lot of ways to deliver, and I think at the core, when you're talking about a nucleic acid, I think the biological precedent is a virus. So we know that viruses are good at this, and we know that there's this what's called the Baltimore classification, which is the classification of all viruses. And you just see it's when I was at my previous company at Sana, we thought about it as almost like a cafeteria where you could say, oh, I could choose pieces of each one of this virus. Now, the danger, of course, is that the human immune system has now built a lot of defenses against this in terms of immunogenicity. And so what we had seen, and this is, probably close to 6 decades of work on lipid nanoparticles, are encasing the nucleic acids instead in chemical entities that we can synthetically generate. And these lipid nanoparticles are safe in the blood. They generally will, we want them to be tolerable and not interact with the blood proteins, and they're going to be in stealth mode, so they don't get recognized. And they then can enter a cell, usually get into the endosome, which I always think of as kind of it's the jail of the cell. It's the place where the cell is sampling. And then our job is to escape and deliver the genetic cargo. So in both cases, they're delivering genetic cargoes. The viral vectors take advantage of what every virus has solved, often in a bespoke manner. So there's many ways that the viruses get in. In the case of lipid nanoparticles, we are using a mixture of different components. The linchpin molecule is this ionizable lipid, and it's the lipid that holds on to the cargo. And when the endosome grabs the lipid nanoparticle, and those of you that are familiar with the endosome biology, it drops the pH as a way to, kind of, in a way, kind of freeze whatever's in there. If it's a virus or if it's something they don't want, the drop in pH generally will kind of inactivate. We take advantage of that inactivation by then disassembling and then fusing with the endosome and then getting the nucleic acid out. So that's kind of an engineering of that system so that we can get that to work. And the lipid nanoparticles have been, remarkably successful with the COVID vaccine. But going forward, the things that made a COVID vaccine good, which might have been a little bit of reactogenicity, so turning on the innate immune system because of some of the components of the LNP, is not something we'll want for therapeutic usage in other contexts. So our goal is to build in two axes. One is tolerability and one is biodistribution. We're trying to make sure that they're tolerable and that we can send them to a lot of places and have them deliver their nucleic acids there.
You presented data at ASGCT on both, I want to say, adipocytes and
Correct.
CD8 T cells. It would be great if you could talk a little bit about what you saw in each of those environments and a little bit about the strategy.
Yeah, so I would say that Mirai's, just to take a step back, Mirai's approaching this in kind of two big areas. One is there is a view in the field that ionizable lipids can not just grab onto the cargo and release them, but they can also provide some of the tropism. Now, admittedly, that's a lot to ask from a small molecule. It's a lipid.
Yeah.
It's got a head group. It's got tails. It doesn't have a lot of information encoded into it. But we have found that approach has been very valuable to get to sites like the lung and the pancreas. And so we take advantage of those lipid nanoparticles where we see some intrinsic tropism to try to understand, how can we leverage that? And one area that we did that in was adipocytes. This is actually a direct injection where you can inject into the adipocyte and then ensure that it doesn't leak out and it stays in that compartment of the adipose tissue. And that was actually done, and I think we might get into this later, but I'll just mention right now, it was done through a machine learning based approach where we had good tropism, but we were able to turn it into great tropism by using ML-based iterative algorithm. So that allowed us to now get molecules and nucleic acids into these adipocytes uniquely after that injection, and then be able to carry out, a number of downstream pharmacodynamic readouts. And we have a number of partners interested in that. So, again, some of this is partner driven. Somebody said, hey, I'd like to directly inject in adipocytes, so we solve that problem.
Yeah.
Maybe the other side of this is T cells, and this is an area that we advanced independently, knowing that there would be partnerships available, but we started the work even without a partner. was we saw that there was a lot of interest in getting into T cells. Here we took a slightly different approach. T cells, there's adipocytes, there's a lot of adipocytes in fat tissue, but T cells almost exist in areas where they're not the only cell type. And
Yeah.
so what we found was that simply finding tropism to a tissue like the spleen or other lymphoid organs wasn't going to get us into T cells. So what we built was a vehicle that would get to, say, the spleen and detarget from the liver because you don't want it going to the liver if it doesn't have to. And then we could add in targeting agents on top of the lipid nanoparticle that then specify which cell type you're going to get into. And so the T cell demonstration is kind of twofold. One is to show that if we deliver a partner's cargo that has a chimeric antigen receptor, we can actually get B cell depletion in
Yeah.
a monkey. And so that was, I think, a exciting opportunity, and we can show that it's also tolerable. So you can get it in, you can get sustained B cell depletion for about a week. And then you can also show that it's tolerable. But it's also a proof of concept that this base LNP that detargets the liver and can be programmed to a T cell could be reprogrammed to other cell types. And so we've shown
Right.
that you can reprogram it to NK cells, B cells. And I think this modular concept sits alongside the intrinsic tropism of the ionizable lipid. These are two different ways to approach designing the lipid nanoparticle and biodistribution for it.
Yeah, I think this is probably a good time to dive into the ML
Mm,
approach. What I'm really interested in learning is like, yeah, what is the starting point? And then
yeah.
how do you manage all of these different potentially competing vectors you're optimizing on? What can you fully predict computationally? Where do you need a wet lab in the loop? Like if you take us into the ML feedback loop, I think that'd be
Yeah,
super cool.
happy to do that. And what I'll say at the outset is, of course, I think what we all want at some point is what I call chat LNP, right? Where you
Yes.
just type in, I want to go here with this potency, I don't want to go there. We're not there yet. And so what we decided early on at Mirai was that the data, if we wanted to build out a strong ML platform, in our hands had to be in vivo. If you want to go extrahepatic, you need a circulatory system, you need a way to biodistribute, you need, kind of to avoid the liver. So you need all of these pieces. And so we decided to, embark on a campaign where we had smart chemists that could define different ionizable lipid kind of chemistries, and those properties then would be tested in vivo to see where they distribute. And that is one good starting point for any place that we might want to go. So we look at all the major tissues. We do this in a mouse right now, and we look at, kind of liver, spleen, pancreas, lung, heart, bone marrow, pancreas, so a number of sites. But the most important thing is we get that view of all of those tissues with each different ionizable lipid. The second feature of that is now that's a base by which to have ML navigate chemistry as well as formulation. I'll just say one word about formulation. These lipid nanoparticles are put together with four or sometimes more components, and the proportion of each one of these is what we refer to as formulation. So the formulation of those and the individual chemistry of each of the elements is something that we can give the machine access to as a lever. And what we'll do is we'll give it this database and then we'll give it an objective and say, listen, I want to go to the spleen, but I want to avoid the liver. So what it's good at is saying, oh, you've got a few good examples here. What I'm going to do is I'm going to push the boundary of those either through formulation or through chemistry. And I'd like you to formulate those for me and tell me how it goes. And so, not to make it sound like it's an ML overlord or anything, but this is a collaboration with the
Yeah.
machine where it then gives us formulations. We formulate them, we put them back in vivo, and now the machine can laser focus on that objective. And then it'll take those answers and it'll say, oh, that's not the right direction to go in. This is the right direction. Oh, I've made some progress here. And the goal is to each time make some progress. And what we found, and this is, I think, perhaps for me was a little bit surprising, is that within 3 or 4 rounds, we're we're at a very high
Wow.
performing lipid nanoparticle. And that in vivo work is done quite efficiently, and the formulation done quite efficiently at Mirai. So we can, 4 rounds is about 6 months. So 6 months to go from kind of something that's good and kind of looks like it gets there, but maybe has other places it goes to something that's selective for that tissue, that's pretty fantastic. And we use that in both the adipocyte work, where we wanted to screen out any of the delivery to, say, a liver or spleen when you inject it in the fat pad, and also for that common base LNP to get it to go to the spleen, but then eliminate the liver. And so both of those were designed, using this ML-based approach. And it's something that, we're really excited about using over and over again. And the other key thing here is, this term, I'm reticent to use the word flywheel because it's overused, but it really is the case that every in vivo study we do goes back into the database.
Yes.
So it's learning and it's gathering data. And even if it makes a bad choice, that might be a bad choice for that objective, but
Yeah.
it could be a good choice for a different objective that we may use down the line.
Yeah, absolutely. Are you able to multiplex them in some way? Like, because I'm imagining you have this when you use LLMs, you can set the heat, which will tell you just how out of the box you want it to think, right? And I
Right.
imagine you probably have some. This is the best. This is the hill climb best choice, but you may want to go left field intentionally to see if there's a new peak.
Yeah, and I guess the term that, kind of ML scientists use is exploitation versus exploration. And so we
Yes.
can tune that in. So exploitation is just looking around things that already work and just fine tuning. And exploration is, like you said, left field. Let's go over there and see what that path looks like. And if we get some traction, we'll go over there and test some more. So we can tune that in. And usually you're doing a lot of exploration early and you're doing a lot more exploitation later on, to be able to fine tune when you've got good nanoparticles.
Are there parts of the process where the computational methods are resisting the biology in some ways, like where it's hard to predict and you need a lot more wet lab data to build up that machine human intuition in some way?
Yeah, so I would say that we are at the early stages of how to turn a chemical entity into like numbers, right? That ultimately that's what the machine deals with. So being able to do that encoding, I think is the term that, we would use in the ML language is I think still a challenge for us, is what's the right way. And as you can imagine, this is again, you mentioned that, kind of I was a systems biologist. I think we call this a multi-scale phenomena, right? You go from a tiny little lipid, it becomes a nanoparticle, it then gets injected into the bloodstream and it's got to get into a cell. So that's a lot of scales for a machine to learn. So what we're trying to do is try and understand how we could, chunkify and, really modularize the study so that we can try to have the machine learn something about the lipid, then have something learn about how LNPs are made, and then something about the in vivo.
Yes.
That's something we're trying to move towards. I think if you had a big enough LLM, maybe people think that you can just, learn everything. This is maybe where my age shows a little bit, kind of feel like I still, at some point, we want to crack the box.
Yes.
If we get good performance out of these kind of multi-objective optimization problems, we'd like to know what it learned. And I
Yes.
think that that's another reason to kind of maybe modularize it. But to your broader point, chemistry and ML, we've seen some great successes. So I don't want to, certainly in the context of proteins. And I think this is an area that, kind of, I think your audience will know something about. But I think when it comes to chemistry, and particularly ionizable lipids and lipid nanoparticles, I think we're just at the beginning. And yeah, it's an exciting beginning and we're making progress, but we're going to need to understand chemistry a little bit better, nanoprecipitation, which is the term given to the formulation approach, and then really what happens whenever you put any body into it's a journey as you go into the blood and then wander around and find the cell of interest. There's a lot of biology that you need to interact with there, and integrating all of those is kind of really the key.
Yeah, and that modularity you talk about, is it kind of literally in the physical geography of the lipid nanoparticle, the locate like when you're thinking about learning something about tropism, for example, which compartment or which organ system it naturally traffics towards. Is there a location on the lipid nanoparticle that tends to drive that, or is it not such a one-to-one mapping in that way?
Yeah, I think there's a lot of I think there's a prevailing theory, which is called the corona hypothesis. So again, as you put material into the blood, blood is full of proteins and, all those serum proteins and other cells. And the coating that occurs as a result of that, I think, is thought to drive a lot of this tropism. So I think that that's where, kind of the goal is to have the outside tell you where to go, and then the inside be protection and the delivery mechanism inside the cell. That would
Yeah.
be the ultimate goal. And again, this is that modularity. You can't have modularity without abstraction. So I would like the outside to just be the directional
Yes.
vector, and then the inside be the one that when you get to your destination, then it takes over to deliver the nucleic acid. That's what I'd like. That's not how chemistry and biology work, but that's what we're trying to design towards.
What actually attracted you to join Mirai in the first place? When was it? What had you thinking about it, and what was the concept at the time? Tell me a little bit about that
Yeah,
decision to join.
if I don't mind, if you don't mind, I'll just go back a little bit farther. Just, I was a faculty member.
Yep.
I was a basic scientist, and we ended up working with a number of companies, and I identified some of the language and processes that they used that they termed drug development, which frankly I had never been exposed to, even as, kind of a more senior person. And so what you do in academia, and you're familiar with this, is you start a course. If you don't know something about it, you start a course. No
It's
better
the
way
best
to
way to learn. It really is
best way to
teaching.
learn.
Yeah.
There's nothing like the, kind of having to show up for students and know a little bit more than they do. That is a powerful motivator. We started a course, and I had a lot of people from the outside world come in, and that turned into a sabbatical for me. And that then turned into the first company that we started, which was Cobalt at Flagship Pioneering, and then merged into Sana. And that was a delivery play, and that was using a viral vector. And I think that having thought about nucleic acid delivery for many years as a basic scientist, it was just a natural way to say the tools that we use for basic science should be the tools of therapy.
Yeah.
And when I left Mirai in 2024, I left Sana in 2024. I found that that itch for in vivo delivery was just something that I had not, and we knew that lipid nanoparticles were going to be a fantastic part of this. And there were things that you could do with lipid nanoparticles that you couldn't do with viral vectors. And so came back to Flagship. The Mirai had been going for a little while already, and I came in and we really tried to reinforce this idea of the in vivo delivery, the ML, and try to really bring that to the table so that we could bring all the modern tools to the in vivo. And LNPs are different than viral vectors, but I think in many ways, they're, I always like to think of it as, the armamentarium. If you're Batman, you have a utility belt, right? You have lots of tools. And so the goal is to have as many tools as possible. And I think lipid nanoparticles have some real benefits that I wanted to be able to try to exploit. And the idea of a focused on a delivery asset only, and then partnering with and so enabling many therapeutics, I think, was kind of a platform within a platform. So the idea is
Yes.
that, you could make one thing that goes to T cells but serve many people who had different cargoes. That was a very exciting prospect as well. So I think we're at the early stages of that, but I think we're very excited about the data we have, and the interest that we've generated.
Yeah, I think the Bill Gates definition of platform, if I've got the history right, which is a little bit more commercial, but is basically about making the people you work with a lot more money than you make yourself. And that's obviously the case with Excel or Word or something like that. But I think and this isn't a comment on the commercial potential, rather the size of the
Right.
market of, if I would imagine the adipocyte work can serve GLP-1s and some of the largest markets on earth, and you can help a lot of people deliver a lot more effectively. Likewise, the in vivo CAR T is just too big of a sector for anybody to do all the work. So being able to enable everybody who's in the space, or a
Yeah.
big chunk of it, is probably a really obvious platform in the truest sense of the word.
Yeah, well, I hadn't heard that definition, but yes, kind of that's that. The goal is certainly to enable others, and I think maybe in therapeutics we might add one component to that, which is, but from our point of view, it also allows us to diversify the bet, as it
Yes.
were, right? So you have T cells, and then the active pharmacological ingredient, as it were, kind of is the nucleic acid. And so we don't know what's going to happen when you get to humans. And there's always a lot of challenges that of biology that we can't model. And so if you now instantiate a large number of people and enable them to be able to put in, again, from a business perspective, now Mirai is placed a lot of bets. And again, to your point, we're seeing some of that come to us. But of course, the goal is to enable partners so that they can be remarkably successful, kind of not just in an HP study, but clinical evaluation and hopefully commercial success. And we do our best to make sure that that's the piece that we can control.
What if you have a collaboration with a drug developer, what is the nature, if in its best sense, what does the nature of that collaboration look like? Because there's probably data they're generating that is really helpful to your process and vice versa. Like how do you run those thread those two processes together?
Yeah, I think, it really depends on how they enter. So, if we have something that I would call cargo ready, one thing that I'll just take a moment. One thing about being in the flagship ecosystem is access to a lot of different nucleic acid cargoes. So
Yeah.
it's not just mRNA anymore. Now every form of RNA is being used in some way to create a therapeutic opportunity. And so we've been able to encapsulate many of those. So, we tend to call the things where we've got a lot of data, NHP, tolerability, a cargo ready vehicle. So there, the idea is that a partner will bring already an established cargo, and our job is to do an evaluation with them, and then hopefully get them enough data to indicate to kind of be have some confidence about nominating a drug candidate. And once that happens, then I think part of our role is to ensure that they can take that material, form a good therapeutic asset with it, help them with manufacturing by tech transferring the the asset into say a CDMO so that they can make it at scale. Along the way, what we've done is we've asked them to take a license to our lipid. So that's really a little bit of the kind of the underlying business model of how we would then, make money. Ultimately, that we have to stay in business. And so keep doing to keep making innovations. We want to be able to bring that money back. And that's really how we enable the partners. On the other end, you can imagine somebody says, "Hey, we really love the tools that you've developed, kind of the machine learning, the ionizable lipids. We want to go somewhere where it doesn't look like you're there, but we think, if we work together, we'll be able to get there." And that's a much more kind of research activity that hopefully turns into ultimately NHP data, which is, some of the most, confidence building data that then goes in there. So a longer path, but I think that we're excited about enabling partners at every road, every part of the development pipe, the early research pipeline, so that they can move on.
Amazing. As we start to wrap up here, what are the big things that are on the horizon for you in the next couple of years? What are you most focused on that we haven't talked about yet?
Uh, so I think, one of the exciting things is, what we've been deeming kind of what we call transendothelial delivery is that, as you put stuff into the bloodstream through an infusion, I think that it's an exciting opportunity to be able to now take advantage of that trafficking system, as it were, the circulatory system, and then be able to exit the highway at the tissues you're interested in. And being able to do that in a manner where we can get to the brain or the muscle, so that we can get to cell types of interest. That's I think one of the areas that we're going to see the most excitement. And we have these partner meetings where we introduce the technology. I often measure it by the amount of time before somebody says muscle or CNS,
Yeah.
right? I think people have really good therapeutic ideas. They're stymied by delivery, and our job is to, kind of lift that barrier. So we're working on that, and I think that's an area where, again, I think we have some biological precedent. There are particles that are transported through the endothelium to the back to the parenchyma behind the endothelial cells, and so we just need to take advantage of that and find that. So that's a very basic science activity that's ongoing at Mirai to be able to break open those tissues. And those unlocks, I think, will, really enable many, many different kinds of therapeutics.
And the reason you're hearing muscle so much is that skeletal muscle, cardiac muscle, is
Correct.
it both in equal measure? Is it more cardiac?
It's I think it's both. Yeah, I
Yeah,
think it's both. I think in the context of cardiac, we see a number of, again, it's all kind of backward from an indication, the kinds of indications that you might want to be able to do where, as something like, you want to make VEGF locally so that you
right.
can get revascularization, say post
Yeah, interesting.
myocardial infarction, all the way to a skeletal muscle where you're talking about Duchenne diseases where you may want to just complement, dystrophin and be able to put that gene back in. So I think there's just, what's exciting for me has been for a while is that delivery. I got to be careful how I say this. I feel like it's future proofed against what comes next.
Yes.
It's going to be something like Cas9 that is going to be equally amazing. We've pulled it from somewhere, some piece of, and it's still going to be encoded with an RNA or DNA, and we can still put it into lipid nanoparticles. So this is, I think, something where if you get the delivery system right, no matter what innovations happen out in the world in terms of therapeutics, I think we can, keep that keep the access open for people. So
Yeah.
I think that's what delivery is exciting for me is that, kind of we can innovate so that enable many different kinds of biology can be delivered.
There's a with viral vectors like AAV, there's a pretty well-known cargo size limit that is very limiting in a lot of these cases. Do you have any analog in LNPs or not so much?
Not so much that we've seen. Again, we've played with some fairly big mRNA, so self-amplifying mRNA, which has been the basis of a recent vaccine. That is about 12 to 16 kb of mRNA, so you can put that in. So I think we haven't seen a size limit. I think the other big thing that comes, of course, is immunogenicity with viral vectors, and
Yes.
we have a good sense that there are varying views of kind of what immunogenicity is going to look like for LNPs, particularly now if we put proteins on the outside to target them. But I think we're going to find out very soon some of that human data, and I think we're all doing our best as a field to try to mitigate that by using humanized binders that look like they're of human origin to mitigate some of the immune response. But
Yeah.
yeah, I still think we need all of these, in the bucket, and so we need to be able to make sure that we can bring whatever is necessary for any particular tissue. AAVs have been really good at getting to the brain from the vascular space. So that's something that we still can't do. So we wouldn't want to, we don't want to take it off the table, certainly.
Yeah, yeah. I mean, you anticipated one of my questions, which was going to be how does this all shake out? And my sense is that you're going to have some things that naturally lean towards virus, some that naturally lean towards nanoparticles, and also on the modality point, right? Not everything is going to be gene editing, not everything's going to be RNA. You, like you said, you have to play for an uncertain future.
Yeah, and I do want to feel like, it's not that we're agnostic, but I think you want to be educated about which one is the right choice, just as you said. And we need everybody to kind of, work together to do this. And I think the real key is how do we all meet down at the regulatory pace? Because if we have so many ideas about nucleic acid cargoes, we should really be generating a lot of therapeutics, right?
Yes.
We should be able to get into people to evaluate and resolve some of their diseases, but we're not seeing it at the pace that we expect. So kind of that's the other place where we want to see more of this stuff. And so you see the two possibilities are COVID, where you have a pandemic worldwide, and then you see baby KJ, N of one type, and we
Yes.
need to figure out how to bridge these so that we can bring that level of scale to even things like rare diseases so that people can have the access to the technology quickly.
Yeah. Well, thank you so much. I've said at the start of the episode that I've been wanting to have a conversation about opportunities in delivery for a long time, and you delivered. So so thank you. I really appreciate you
Well,
taking the time.
yeah. Well, thank you for having me, Patrick. This is a wonderful opportunity, and yeah, excited about what's going to happen in the field.
Likewise, thanks so much, and everyone, thank you for listening. We will see you next time.
Thank you very much.
Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sanogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sanogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.